When we are infected with a pathogen, such as a bacteria or virus, our bodies respond by fighting the infection through the immune response. The immune response to infection differs according to the type of pathogen that the host is infected with. For example, the immune response will be different when a host is infected with a viral pathogen compared to when they're infected with a bacterial pathogen. The field of proteomics studies the full set of proteins present inside an organism, whilst the field of transcriptomics studies the full set of genes expressed inside an organism. When an individual is infected with a pathogen, there will be changes in gene expression and protein production as part of the immune response, and we can see these changes when we look into the transcriptomic and proteomic datasets obtained from these individuals. In fact, there will be pathogen-specific patterns present in the host transcriptomic and proteomic datasets, and we use these patterns in our research. One example of how we use these patterns is through identifying gene or protein biomarkers of infection. When a child has fever, it can be challenging for doctors to differentiate between a fever caused by a bacterial infection and one caused by a viral infection by studying the clinical symptoms alone. Normally, the child will be infected with a self-resolving viral infection that doesn't require antibiotics. However, sometimes they'll be infected with a life-threatening bacterial infection. This means that many children with viral infections are often given antibiotics unnecessarily, while serious bacterial infections can occasionally be missed, with potentially fatal consequences. We apply feature selection methods to transcriptomic and proteomic datasets obtained from children with bacterial and viral infections to identify biomarkers that we can use to accurately diagnose bacterial and viral infections. The aim is that doctors will be able to use these biomarkers in a rapid diagnostic test to more accurately differentiate between bacterial and viral infections in children. This is just one example of how we can use host omic datasets in the context of infectious diseases. Omic datasets can also be combined using multi-omic integration. This is a powerful approach to studying omic data since it provides a more complete picture and it enables you to model the interaction between different molecules